Okay. So it looks like we're ready to talk about the supporting cells of the nervous system. Last time we met, we talked about neurons, and we learned that they are the kind of major functional cells of the nervous system, generating and conducting nerve impulses. Learned about their structure, the dendrites, the cell body, the axons, axon terminals, synaptic end bulbs, all that wonderful stuff. And we talked about the different ways to classify them structurally and then functionally. Okay, so what we'll talk about here today, first thing, we don't really talk much about these other than really just these couple slides and really kind of a couple times. But they are important to talk about the supporting cells. Okay, neuroglia, also just called glia, are described as non-excitable, which means that they are not involved with creating and conducting nerve impulses. Okay, they don't use ion flow to perform action potentials like we'll talk about here with neurons. They might help neurons do that, but they're not the ones that are conducting the impulses. Okay, there are 6 total neuroglia. 4 of them are in the central nervous system, and that's what these 4 here are on this page. The first are called astrocytes. They're the ones here in kind of the bluish purple. As it says, they wrap around blood capillaries, like this blood vessel that you show right here, and they contribute to what's called a blood-brain barrier. Blood-brain barrier is exactly what it sounds like, abbreviated BBB. It's a barrier between our blood and the brain, or if we're looking at the spinal cord, same thing. They can control what can leave the blood to enter the interstitial space between the neurons. Okay. So they control and prevent things like toxins and drugs and that kind of stuff from traveling through. And we'll talk more about them when we talk about the brain. And they also pick up nutrients from the blood and help pass it along to those delicate cells, the neurons, that is, of our brain and spinal cord. They also, as you can see, this one right here is supporting neurons because they have considerable strength. Microglia are the ones here in green, are the immune cells of the 4, responding in areas of infection, trauma, or unfortunately stroke, where they function as phagocytes, which means they are like the Pac-Man I've been talking about, running around chewing stuff up, digesting them, things like cell debris, microbes, damaged nervous tissue. Ependymal cells are the ones that are down here at the bottom, kind of cube-shaped or columnar-shaped. A lot of them have cilia or microvilli, which is to help with secreting, but then circulating what's called cerebrospinal fluid, the fluid that is found in places like the ventricles, which are chambers in our brain, the central canal, which is a little tiny chamber inside our spinal cord. We've mentioned cerebrospinal fluid before. We'll talk more about it here in our next lecture. And then probably the coolest ones, as far as name goes, would be these that kind of look like an octopus, what we call oligodendrocytes, which, as you can see, have little leg-like extensions that wrap around the axons of neurons, creating what's called a myelin sheath, a multi-layered lipid and protein covering around some axons, insulating them and increasing speed of nerve impulse conduction. Okay, so the yellow cell here is the neuron. The pink shaded oligodendrocytes wrapping around the axon, but not completely. As we can see, there's these spaces with the neuron, the axon of the neuron, I should say, that's uncovered, what we call a node of Ranvier. We'll talk a little bit more of that here on this page. Okay, so those were the central nervous system neuroglia. There's also the peripheral nervous system neuroglia. There are 2 of those, Schwann cells and satellite cells. Schwann cells are cells that wrap around a single nerve fiber, creating several layers of fatty insulation. Again, same thing, called the myelin sheath. Same thing, it protects the nerve fibers, electrically insulates them from one another, and increases speed of nerve impulse transmission. Let's talk about the microscopic gap, what we call the node of Ranvier. Okay. Now this is showing you a picture of an oligodendrocyte. The same thing would be the case if we were looking at if it was a Schwann cell. Okay. What these nodes of Ranvier allow for is an actual potential nerve impulse to skip from node to node. Think of it like a rock skipping on water, which enables a signal to travel at a really rapid pace. So that's why the myelin sheath increases speed of nerve impulse conduction. And we'll be talking more about that here later in this lecture. What's cool about Schwann cells, the outside layer of them is a wrapping called neurolemma, and is essential to regenerating injured nerve fibers, which is something that we don't really do so well in the central nervous system, but can do in the peripheral nervous system because of that neurolemma. And that explains why, you know, if you have brain damage or spinal cord damage, typically it's pretty much permanent. And whereas if you get like nerve damage out in your arm or leg, you can a lot of times fix that because of the axon regeneration that the Schwann cells perform. A difference that exists between Schwann cells and oligodendrocytes. Oligodendrocytes wrap around and create an actual potential, or excuse me, myelin sheath around multiple neurons, like this one here, that's wrapping around 2 different neurons. This one's wrapping around these 2. Schwann cell, as it states, only wraps around a single nerve fiber. So it's only going to wrap around 1 axon. Satellite cells, we don't really talk much about those, but they surround the cell bodies. Remember, the cell bodies of a neuron are these parts here. And they support neurons by regulating exchange of materials between the cell bodies of those neurons and interstitial fluid. We don't, like I said, talk much about those. The main thing we do talk about as far as these cells go is myelin. Okay? The myelin sheath formed by oligodendrocytes in our central nervous system, and the myelin sheath in the peripheral nervous system formed by the Schwann cells. Now, not all nerve fibers, or in other words, not all axons are associated with myelin. And if you were to dissect the brain and spinal cord, what you would find is that it looks like there's kind of more lightly colored areas and more darkly kind of gray colored areas, okay, which we literally call white matter and gray matter. Okay, and the white matter in our cerebrum, the biggest part of our brain, is kind of on the inside, whereas it's the reverse in our spinal cord, it's on the outside. And it is white in appearance because there's an abundance of myelin. Okay, the myelin sheath, that fatty white color, is what causes it to look white, whereas gray matter in our cerebrum is on the outside, forming a thin shell. It forms a capital H on the inside of our spinal cord. Okay, and this is gray in color because there's very little to no myelin, which is because instead of finding myelinated axons, instead you find the cell bodies. Remember, cell bodies of a neuron would be this part, the dendrites. They don't have myelin sheath, nor do cell bodies. You also find the axon terminals. There's only myelin in this area of a neuron. So if you have a collection of things like this, you're not going to have myelin. Okay? Now, not all neurons are going to be myelinated, so there are bundles of unmyelinated axons in there as well. We'll talk a lot about the gray matter in our brain. This very thin shell of our cerebrum is called the cerebral cortex, which is where the highest level processing thinking takes place. Things like the ability to understand the words that I'm speaking right now, the ability to see and process what you see takes place in our cerebral cortex. Process what you taste, what you smell, what you feel. So those are all sensory things. It's also involved with your personality and your conscience and things like that that are unique to each and every one of us. It's also where signals arise and begin that travel down and cause our muscles to contract. So we'll be talking in our next lecture about what we call functional areas of the cerebral cortex. It's very amazing. This is only like 2 to 4 millimeters thick, so super, super thin. Super, super amazing though. We're not quite ready to talk about that though, because we still got a lot more to talk about in this lecture. And let's let's talk more about neurons and the ability to be electrically excitable. Once again, which means that they respond to a stimulus and they can create one of two signals, either a graded, also known as a local potential, which is used for short distance communication only. We'll talk about that first, and then we'll get into much more complicated action potentials. Another term for action potentials are nerve impulses to allow communication in our body to occur over long distances. So like, carrying an impulse for the sensation of itch from the bottom of our toes all the way up towards our brain. In living cells, the flow of ions constitutes the electrical current that flows down neurons. Graded potentials and action potentials occur because the membranes of neurons contain many different kinds of ion channels that open or close in response to specific stimuli. So here on the next couple slides, we'll talk about these ion channels and how they allow ions to flow in or out of a neuron, which changes that resting membrane potential that we talked about last week, which we learned in most cells it was negative 60. In neurons, we'll see that it's a little bit different than that. And when ions flow, that number changes, and that's what we'll be talking about. That's what signaling in our neurons is all about. Okay. It's kind of, I said the next slide, I guess I lied. We'll talk about the channels here in a second, but here's an example of something that you guys have been doing, taking notes, right? So when you put a pen or pencil in your hand and you squeeze with your fingers, you have sensor receptors that are sensitive to that touch, okay, that become excited because there's your stimulus, the touch, which triggers a graded potential, okay, in a sensory neuron or a sensory receptor, which you can see is down in the finger with the number 1 attached to it, okay. The graded potential triggers axons of the sensory neurons to form an action potential, which then travels along the axon into the central nervous system, ultimately causing release of neurotransmitters at a synapse with an interneuron. So we create, graded potential, which then triggers an action potential that travels all the way up and into our spinal cord. We actually travel up the white matter of the spinal cord towards our brain, where we interact with a second neuron, what we call the interneuron. Synapse is where these two neurons meet, triggers a graded potential in our second neuron, which triggers an action potential, which then travels up. We see another neuron that carries that signal up to our cerebral cortex, where perception occurs. Remember, perception we talked about last time was where we had that conscious awareness, where we can feel that touch of the pen, or we can feel that itch, that tickle, that kind of stuff that we talked about. Okay. So now we want to control our pen and write. So we initiate a signal from a different area of the cortex down a motor neuron that travels down from our brain through our spinal cord. We have a synapse, a graded potential occurs in the second lower motor neuron, and then an action potential travels out to our skeletal muscles, which then we release chemicals called neurotransmitters that cause our muscles to contract in a specific coordinated manner, allowing you to write and make your notes. So something as simple as just writing, putting a pen in your hand and using it, look how amazingly complicated that is. Now we haven't really got into what all goes into each of those graded and action potentials, but we're getting ready to. Because I stated, it all relates to resting membrane potential and the flow of ions. As we learned, resting membrane potential is the excess negative charge on the inside of the cell, what we call it cytosol at rest. In other words, at an unstimulated state. At rest, neurons have a resting membrane potential of negative 70, which is a little bit more negative than the negative 60 that we said most cells were at. Okay. The following are ion channels that can open, allowing specific ions to move across the plasma membrane down their electrochemical gradient, and the flow of these ions through these channels can result in a change to that resting membrane potential. The first, shown at the top, are called non-gated or leakage channels, all the same thing, which is it states there in the picture. I know it's kind of small, but hopefully you can read it okay. The channel randomly opens and closes. And this one is an example. If that little box will go away. Potassium, which we can see is a lot concentrated on the inside of the cell, very little outside. So when that channel opens up, potassium rushes out down its gradient. Okay. Ligand-gated channels open and close in response to a specific chemical stimulus, like a neurotransmitter, a hormone, and particular ions, which can lead to a neuron becoming excitable. In this case, we see a chemical, doesn't matter what that is. It's labeled as acetylcholine, which we'll talk about, but it could be a hormone, whatever. Binds to the channel, which is closed without it. So the chemical acts like a key to unlock the door and open it up, allowing the ion to flow down its gradient. Okay. Mechanically gated channels open up in response to a mechanical stimulus. Things like vibrations, like sound waves in your inner ear, touch, pressure, or a tissue stretching. Okay. So when you push it on your skin, for example, you're going to trigger these types of channels to open up in certain neurons. The channel opens up, the ions flow through. The ones that we're going to talk the most about in this lecture are voltage-gated channels, like this voltage-gated potassium channel, which you can see at -70 at rest, because we just learned that's the resting membrane potential of a neuron. -70, the channel's closed. But here, when the voltage is at -50, the channel is open. So that's what they do. They open in response to a change in membrane potential or voltage. And the reason that we talk about them is they are important for generating and conducting action potentials. Table 12.1, I included it. It was in your textbook. It just gives you a description, which I just did on the previous page. It also shows their location, which not super important at this time. Now, you might be wondering why the resting membrane potential is a little bit more negative in neurons than it is in other cells. Okay, and that's because there's a small buildup of negative ions in the cytosol along the inside of the membrane and an equal buildup of positive ions in the extracellular fluid on the outside. Three major factors contribute to this resting membrane potential -70. There's a very large unequal distribution of ions in the ECF and cytosol. ECF, remember, is extracellular fluid, so that's outside the cell. It's rich with sodium and chloride. Cytosol is full of potassium and negatively charged phosphates. Remember, ATP is short for adenosine triphosphate. Phosphates have multiple negative charges, and we've got 3 of them in the single ATP. So each ATP is really negatively charged. And proteins also a lot of times have a negative charge, as you can see here on this next page. Okay. The second reason is because of the fact that most anions, which remember are negatively charged, cannot exit a cell because they're attached to something that's non-diffusible like ATP and large proteins. So the things that are negatively charged a lot of times are stuck inside the cell, meaning that they can't escape. And then the third thing is the action of the sodium potassium ATPase pump, which, as we learned, pumps 3 sodiums out of the cell, 2 potassium ions into the cell. So we lose 3 positives, we gain 2. So for each round of the sodium potassium ATPase pump, we lose a +1 charge. As you lose 3, gain 2, 3 minus 2 is 1, right? Okay. And so moving more cations out of the cell than anions into the cell further increases that negative charge. Questions on that? Not super kind of important specifically, but this explains why it's just a little bit more negative, that -70, than what we saw in other cells. All right, let's talk about our first electrical signal, what's called a graded or local potential, which we said is a small change away from the resting membrane potential. And we call it local potentials because it's typically happening in a very specific or local region in the neuron's plasma membrane. So You know, looking at our neuron here, maybe it's just like right there at the very tip of that dendrite. We place an electrode there, okay, we might see a change, very small, occurring in that specific spot, okay? There's two types of graded or local potentials. One that causes the number inside the membrane to become even more negative. One that makes it become less negative. Okay. The response that makes the membrane more polarized, polarized in this case means more negative, is called hyperpolarizing. That's what this one is right here. Remember, hyper means high, and we just said polarized means more negative. So we're high negative, more negative, in other words. Whereas a depolarized, and we talk more about these than these, look at our number, it goes from -70 to -60, which we can all hopefully agree is less negative, right? Okay. And so when the response makes the membrane less polarized or less negative inside, we call it a depolarizing graded potential. Okay. A graded potential occurs when a stimulus causes a mechanically gated or ligand gated channel to open or close in an excitable cell's plasma membrane. So we just talked about these channels. Okay. One of these two is typically involved in a graded potential. Okay. We were to zoom in at the dendrites here, or the cell body, maybe we find a whole bunch of the mechanically gated channels, or maybe we find a whole bunch of the ligand gated channels. Some chemical binds to it and it causes something, in other words, an ion to flow in or out. Okay. Mechanically gated channels and ligand gated channels can be present in the dendrites, the branches, as we know, of our neurons, or they can also be found in the cell bodies of interneurons and motor neurons. Remember, the cell bodies where the nucleus is. Graded potentials occur mainly in the dendrites and cell bodies of neuron for that reason. And what's cool about them is they can actually add together. But this all has to occur as a response to a stimulus. And stimuli can include like chemicals, whether it's a hormone. When we taste something, when we smell something, there's chemicals involved with that. Mechanical could be like touch or stretch, as we've talked about. Heat, like when we feel the warmth in our skin from the sun as we walk outside. And then light, that's what we are seeing when we have vision. Okay, here's an illustration of those channels opening up. In this case, the first one is a pressure sensitive mechanically gated ion channel. Okay, it's labeled as a depolarizing, which means we become less negative, right? Look at the inside of the cell here at rest. It's negatively charged with the negative symbols. Pressure is applied, the channel opens up. Maybe it's sodium rushes in, I don't know, it doesn't matter at this point. But look what's happened on the inside of the cell. It's become partially positive. Okay. Here, letter B, this picture shows a chemical. Looks like it's acetylcholine. A neurotransmitter binds to that channel. It's like a key, unlocking it. Same thing, depolarizing. We become less negative inside. In this case, actually becoming positive. Okay. And then this last one is a hyperpolarizing graded potential, which means that as we see chloride's going to rush in. And chloride, as we know, is negatively charged. If we bring a bunch of chloride in where it's already negative, will become even more negatively charged. So we dip down instead of going up and become more negative. Does that make sense? Clear as mud, right? Here's another example. We're showing, looks like a bipolar neuron. We're zooming in at this little segment of its dendrite. We see it's got a ligand-gated channel. Here's a ligand binding to it. Triggers the flow of sodium in. So this would be an example of a depolarizing graded potential. Because sodium rushes in, the positives make us become less negative on the inside. Or you could also say become more positive if you are I guess a glass half full instead of a glass half empty kind of person. Now, we can say that electrical signals are graded, which means that they vary in amplitude or size, depending on the strength of the stimulus. And that's what we're showing off with this kind of graph on the top right. Okay, hopefully you guys can all see with the kind of second part of the picture, stimulus 1, which is pretty weak, because that's what we're measuring here in the y-axis. Stimulus 2 is a little bit stronger. Stimulus 3 is the strongest. Everybody agree with me there? Pretty straightforward. Look what happens to resting membrane potential for those three things. Stimulus 1 causes a very small change. Maybe we go from negative 70 to negative 76. Or excuse me, negative 66. Okay, I went the wrong way with it. The stronger stimulus makes us get to maybe negative 61. Even stronger stimulus makes us get to like negative 58. Okay. What is different is the number or how many of the ligand gated or mechanically gated channels open. So in other words, more of these will open with a stronger stimulus, which means more ions flow through, and more ions flowing through change the resting membrane potential more, right? So there's more doors, there's more ions that can come in or out, doesn't matter what we're talking about now. And the opening or closing of these ion channels alters the number of specific ions that can move across the plasma membrane to cause a greater potential. The amplitude, which is a measure of how much of a change, can vary from less than 1 millivolt to more than 50. After it's generated, a greater potential spreads along the membrane in both directions from the stimulus source. And this is accomplished by what we call local current flow. So look what happens. We depolarize, but we don't just stay at that number of negative 50 whatever or whatever that number is, because we end up returning right away. That's because what happens is the ions flow in. Okay, and remember, if we think back to our chemistry lesson, we said that opposites are attracted to one another, right? Okay, well, if we have a bunch of positives coming in one specific spot, and they're hanging out here, there's a bunch of negatives nearby, guess where those positives are going to want to go? To the negatives, right? Because they're attracted to them. So when those ions move, it's like if you spray Febreze in one area of your room, it's really strong in that area, but then over time, what happens? It moves it out, right? The ions flow out towards the other areas, and we return back to resting membrane potential. So local current flow refers to the passive movement of charges from one region of the membrane to adjacent regions due to differences in membrane potential in these areas. Make sense? Okay, let's talk about summation. As a graded potential spreads out to adjacent regions of the membrane by local current flow, it gradually dies out because its charges are lost across the membrane through leak channels. Okay, we call that decremental conduction. Okay, mode of travel by which graded potentials die out as they spread along the membrane. Okay, so this is why we depolarize, but then we just return. Depolarize and then return. The change dies out. That's what decremental conduction is. Although an individual graded potential undergoes decremental conduction, it can become stronger and last longer by summating with other graded potentials. The beginning of the word summate spells out the word sum. Lauren, you were talking about math earlier and how you talked about a meme or something you saw over the weekend. What do you calculate in math if you're calculating sums? Addition, exactly. You're adding things together. Okay. Summation is the process where we add graded potentials together. Okay. So what this means is, if we look at stimulus 1, stimulus 2 here, let's just say we zap it. We'll call it a zap. Okay. We stimulate the neuron, causing it to depolarize, meaning that we become less negative, as it's shown here. And then right as we're starting to return, look what we do. We hit it with another zap. Okay, instead of stimulus that has the same strength, because these are actually the same strength stimuli, if you add this on top of this, that's what we're doing. Okay. You hit it with a second stimulus before the first stimulus dies out, we end up having an even stronger depolarization. And this is important because this is going to lead us to the steps towards an action potential. Now, what we'll talk about in this lecture are postsynaptic potentials, which involve neurotransmitters being released, and these occur between the dendrites of cell bodies neurons when those neurotransmitters bind to them. We'll talk about receptor potentials and generator potentials in our next unit, actually, when we talk about sensations, because those are involved with sensory receptors and sensory neurons. Okay. All right, now we're ready to talk about action potentials. Also known as nerve impulses, are brief but large electrical depolarization and then repolarization of a neuron's plasma membrane caused by the opening of voltage-gated ion channels. Unlike local or graded potentials, remember those are the same thing, an action potential can travel along the membrane away from the site of origin by a process called propagation. And it all relates to a term called threshold. Okay. Reading through, I know it's kind of cut off, but that's okay. I can still tell you what it says. It'll go away. I can still read part of it. And an action potential will occur in the membrane of the axon of a neuron when depolarization reaches a certain level termed threshold. Okay. So what we have here are three different types of stimuli: a subthreshold, a threshold, and a suprathreshold stimulus. Okay. In other words, this is a really weak, this is a little bit stronger, this is a really strong stimulus. Okay. If we're looking at our first, what we call the subthreshold stimulus, which is really weak, we trigger a depolarization to occur. But we don't reach this dotted line, which is the place where the floodgates open. Okay. We have to reach this number, -55. That's the key, because without hitting that, an action potential is not going to be created. Think of this like the point where we pull the trigger. Okay. We're firing a gun. We can't fire it until we hit -55. Because we don't depolarize far enough to -55, we return to resting membrane potential. Sub means below, right? So a subthreshold stimulus is below threshold. Okay. A threshold stimulus is a stimulus just strong enough to get us to threshold -55. Look what happens when we hit -55. Boom. Something big happens. We see a huge change in resting membrane potential, but then we return really quickly. Now remember, all this stuff is happening in milliseconds. This is like the snap of a finger, super, super fast. Okay. Look what happens with a suprathreshold stimulus. An even stronger stimulus. We hit threshold really easily. Boom, we fire our action potential. Now you guys all agree with me. Everybody look at the graph up here. This is not as strong as this, right? Okay. We'll look at this one. This one was weak. We depolarized a little bit. This one was a little stronger. We depolarized a lot more. It would make sense that this one, which is a lot stronger, we would go past +30. meaning we would depolarize a lot more, somewhere, maybe positive 60. We don't, okay? An action potential tops out at the same point, somewhere around positive 30, 35, no matter how strong the stimulus is, if we hit threshold. Okay. What happens is we fire one action potential, and if we have a super strong stimulus, we'll end up firing more than one continuously. Okay. So each action potential caused by a suprathreshold stimulus has the same amplitude or size as an action potential caused by a threshold stimulus. The greater the strength of the stimulus above threshold, the greater frequency of action potentials until we reach a maximum frequency. Frequency refers to how quick these are, or in other words, how close together they are. Okay, the stronger the stimulus, the closer these will become. Okay, if this was a little bit weaker, these would be spaced out a little bit more. And that's something that we'll talk about in our next unit, and explains why, for example, when someone touches us really lightly, we can tell that's different than if somebody pushes in and really grips our arm or whatever. We can discriminate between a light touch and a very firm touch. Okay, it's all about the number of action potentials in that case. Now, something I want to talk about real quick is the all or none principle, which states that if a stimulus depolarizes a neuron to threshold, the neuron will fire at maximum voltage. There's the all. But if we don't reach threshold, the neuron's not going to fire at all. And there's the none. So either a neuron fires or it doesn't. If we pull the trigger, we fire. If we don't pull the trigger, it doesn't fire. That's why I use the gun analogy. Not that I'm, going to get into gun talk in this class. Okay, so now we know how to get to an action potential. What I want to do for the next couple of slides is talk about the steps and the processes that go into one complete action potential. This is a figure from a different textbook, and I really like it because it gives you numbers over there on the right, which is the steps involved with an action potential. It's from a different textbook. This is the one from our textbook. I just don't like nearly as much. I'll still talk a little bit about it, but I'll kind of focus on this one first. And so here we are. We're measuring resting membrane potential at -70, so our neuron is at rest. Okay. Some type of local or graded potential causes us to depolarize. Whether that's a chemical signal, mechanical, whatever, it doesn't matter at this point. Okay. All that it matters is, did we have a strength that caused us to hit threshold? If we hit threshold, remember, we fire the action potential. And step 2 is where we hit -55 and we go through what's called the depolarization phase, where we open what are called voltage-gated sodium channels, allowing sodium to rush in very rapidly. Okay, remember, we're dealing in here, it doesn't say it, but it might be a good idea to write. This is ms, milliseconds. Okay. We rapidly depolarize, we actually end up becoming positive inside our neuron. So much so that we top out somewhere around positive 30, 35. Okay. But then you can see what we do next. We return right back down into the negative numbers. Those voltage-gated sodium channels that are open here are closed by the time we reach step 4. So no more sodium rushing in. And then step 5, as it says, we have slower potassium channels that open. And these are called voltage-gated potassium channels, which allow potassium to rush out. Potassium is positively charged. If potassium leaves the neuron, we lose the positivity, we return back to negative. numbers. So think repolarization, we return to negative, because that's literally what repolarization means. Okay. Now look what happens here with step 6. We actually dip a little bit past negative 70 because we end up allowing a little bit more potassium out than the sodium that we let in. This is a very short stage that's called the hyperpolarizing phase or hyperpolarization phase, which doesn't last very long because it doesn't matter. By the end, we return to resting membrane potential. Potential, not potential. That's not a word. Potential. These 7 steps will, without a doubt, be very important to know for your exam. I can tell you right now, you might even see this graph not labeled as well as it is, but I might ask you on the exam to tell me what's taking place in step 3 or step 5 or step 6, whatever. So make sure you can, kind of go between the two the picture and then the stuff on the left and the notes, and you can explain it. Okay. Maybe put it on there. Maybe I won't. I don't know. I haven't decided yet. Without a doubt, you will need to know the steps. Here's my advice. Study the steps and study the steps and study the steps. When you feel like you know them, take out a blank piece of paper and see if you can write them out. You aren't going to have to do this on an exam, but if you can do that before the test, you'll be in good shape. Because what I might do is I'll give you like 3 or 4 steps and I'll say what happens next. Or I might give you 4 or 5 steps and I say might number them from 1 to 5, put them in order. So there will be several questions where I want you to know what's taking place during an action potential. I promise once you sit down with it.
What would be the name of step 4?
What's up?
What would be the name of step 4?
I mean, there's not really a name for it. Once you sit down with it, it won't be that bad. Now, this is the figure, like I said, that's taking from your textbook. Same thing, a stimulus causes us to reach threshold, which means we depolarize, and you can see it's shown in green. Voltage-gated sodium channels are open, sodium rushes in, they then close. Voltage-gated potassium channels open, potassium allows for us to repolarize. We eventually hit resting membrane potential, but we continue. Here it's labeled as the after hyperpolarizing phase, okay, where we are even more negative than we were at the beginning. But once again, milliseconds, we return to resting membrane potential almost instantly. Okay. Now, keep in mind, action potentials, if you look at my figure at the top of our neuron, they originate right in here in the axon hillock, specifically the trigger zone, and then the action potential is going to spread all the way down to the tip of each of our neurons, to the axon terminals, specifically the fattened synaptic end bulbs. So this is taking place right here, and then we're going to spread that action potential down, which means we're going to have this segment all the way down to this segment going through a depolarizing phase, a repolarizing phase, all that stuff. And once again, it all involves the flow of ions through kind of 2 main ion channels. A voltage-gated, or a bunch of voltage-gated sodium channels and a bunch of voltage-gated potassium channels. At rest, as you can see, number 1, all the channels are closed. Okay, but when we hit threshold, those voltage-gated sodium channels open up, sodium rushes in, and we depolarize. Then those channels close and the voltage-gated potassium channels open, allowing potassium out of the cell, which causes us to repolarize. In other words, return to resting membrane potential, in which we then
close them off and start the whole process over. All right, let's watch this video.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell.
Okay, so they're just telling you why it's at -70 at rest.
Making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body.
Okay, real quick. What these little kind of lightened areas are showing you, each of those is one of those little tiny graded potentials, a small change to that resting membrane potential, okay? When you stimulate a neuron, like if I were to push in on your skin or push in on my skin, you're not going to end up just stimulating one dendrite. Because remember, cells are really small, but dendrites are many, okay? By pushing in on one small area, you're going to still end up stimulating a whole bunch of dendrites, which means this one will have a graded potential, this one will have a graded potential, this one will have a graded potential. All of those graded potentials, remember, can add up. Summation, right? Okay, we're trying to add up towards threshold. So what they're doing is they're collecting right here where that trigger zone is.
Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered.
Meaning if we get
Action
threshold.
potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse.
Okay, I'm going to stop here and we'll get to that part of the video in just a minute. If I go back to this slide here from your textbook, the figure here on the left where it's color coded is kind of handy
because if you see what they have for green and tan, they have bracketed off as what we call an absolute refractory period. And then here in this kind of darker tan, we'll see orangish color, they have listed as its relative refractory period. Let's talk about refractory periods, absolute and relative, here on this page. During an action potential and for a few milliseconds after, it is difficult or impossible to stimulate that region of a neuron to fire again. We call this period of resistance refractory period, or its refractory period. So I always use an analogy of jumping up and then landing on the ground. Okay. Which, you know, if you kind of think of it, it kind of looks like a jump up and down with a graph of an action potential. Okay. An absolute refractory period lasts from the start of the action potential until we hit resting membrane potential -70 again. So once the action potential is fired, until we hit -70 again. Here's what you can think of for absolute. Think absolutely not. In other words, absolutely no stimulus, no matter how strong it is going to trigger a new action potential during this time. Okay, we are not video game characters. We can't jump a second time until what? We land and finish our first jump. Okay? So if you're jumping up in the air, you can't jump a second time until you land on the ground again. Okay. Now, the relative refractory period is the point from where we hit -70 to the point where we hit it again, because remember, there's that point in time, once again, this is all short amount of time, milliseconds, where we're extra polarized, we're hyperpolarized, we're farther from -55. And during this period, it is possible, but the key is, and I would highlight this, only with an unusually strong stimulus. So think about it, like when you jump the first time and you land, typically our body doesn't just like bounce, it's more like on a trampoline. We jump, we land, it takes us, you know, milliseconds to gather ourselves, bend our knees and jump again, right? This period of time where we are gathering ourselves and jumping a second time is like the relative refractory period. Does that make sense? Lauren, does that make sense?
Yeah.
So even if we have a stimulus, like if I were to walk over to you and push in on your hand, push in your arm, whatever it is I'm doing to you, okay, neurons are firing, but neurons are not firing continuously. Because neurons fire and then they take a short period of time until they can fire a second time. Because you know what? They have to finish the first firing. And then they have to finish the second fire. And then they have to finish the third before they can fire a second, third, fourth, fifth time. And that's why if I go back to this figure, even though the threshold or super threshold stimulus is super strong, we fire, we return. We fire, we return. We fire, we return. Okay. What we have are so many neurons that neurons are firing while others are in their refractory period. So it's like a stimulus is always being there if it's stimulating us, because we're not just involving one neuron ever, really. And we're kind of focusing on one neuron, but remember there's thousands upon thousands upon thousands. Brain has millions and millions and millions. The eyeball, small structure, millions upon millions of neurons. All right, let's talk propagation. For neurons to transmit signals, a transient depolarization and repolarization occurs during an action potential, and it must travel from where they arise at the trigger zone all the way to the axon terminals. This conduction of an action potential along the membrane is called propagation, and the type of propagation is determined based on the type of nerve cell. Okay. So what that sentence or those sentences say, I guess it looks like 2 or maybe 3 sentences. An action potential arises here in the trigger zone, then it travels all the way down to the very tip of each of these branches. Okay. What we're looking at, A, B, C, and D, are the same segment, just that little tiny segment at different points in time. Okay. So this happens first. This is a couple of milliseconds later, a couple of milliseconds later, a couple of milliseconds later. Once again, all super, super short amount of time. Okay, we've got it color coded. I really like this figure for that reason. We see purple represents that this segment is in resting membrane potential. Whereas this very small segment is in its depolarizing phase. Sodium is rushing in. Okay, sodium's rushing in. Remember, sodium with its positive charge isn't just going to stay put. Some of that sodium is going to trickle over here, right? And if this segment reaches threshold, because of the sodium that's come there, that segment is going to end up depolarizing due to opening of voltage-gated sodium channels. So the movement of sodium from one segment to its neighbor causes this segment to hit threshold, causing it to depolarize. And then sodium continues moving down, and this one hits threshold. And then it moves down and it hits threshold. Now look what happens behind the depolarization segment here in green. In blue, we show the repolarizing phase. Remember, the repolarizing phase is where we're going back to -70. So that very first segment is starting to reach threshold. Or not threshold, excuse me, return towards resting membrane potential. And then this next segment will start to return. And then the next segment will return. The pink shows you that the segments each all go through that hyperpolarization phase. Eventually, the first segment where it all began has reached resting membrane potential. And then that one will reach resting membrane potential. And that one will reach resting membrane potential. So every step that we just talked about takes place in every segment of an axon and its terminal branches. Now what's cool about that, everyone look up here, watch me as I show this. So now our action potentials may be here. Back here, we've now seen that we are in rest. So we might have an action potential traveling down here. At the same time, we've got a new action potential. So what a single neuron can have are multiple action potentials traveling down the axon at the same time, just at different points. Does that make sense? And the more you have, the more signals get transmitted to whatever's being stimulated, whether it's another neuron or a cell or a gland or a muscle or whatever. Actually, I didn't read this. Let me read through this. So in a neuron, an action potential can propagate along the axon away from the cell body only. It can't go this way. It only goes this way. So it cannot propagate back toward the cell body because any region of the membrane has just undergone an action potential and is temporarily in the absolute refractory period and cannot generate another action potential. should realize that it is not the same action potential that propagates along the entire axon. Rather, the action potential regenerates over and over at adjacent regions of the membrane from the trigger zone to the axon terminals. So each segment creates its own action potential, which is cool. Two forms of propagation exist, and it all relates to myelin, which we learned earlier wraps around the axon. And those two forms are called continuous conduction and saltatory. Continuous, think of it just like burning a fuse, like lighting a firecracker and watching that fuse burn down the end. And this is what happens, and I would highlight this. Unmyelinated axons is also what we see in muscle fibers as well, muscle cells. But for now, we'll just focus on unmyelinated axons. Whereas saltatory takes place, and I'll highlight this, along myelinated axons. I would also highlight that this is much more rapid than continuous. In this process, like I said earlier, it's like skipping a rock on water. An action potential can skip from node to node to node. These are the same neurons. These three here just at 1 millisecond, 5 and 10 milliseconds. Here's the same one for 1, 5, 10. Look how far the action potential has spread in this one at 10 milliseconds versus here. It's almost at the end of the axon here, isn't it? So our signal's traveling much more quickly here than it is here. Okay. Does that make sense? So the amount of myelination is one of three things that affects an action potential propagation speed. AP is short for action potential. If you didn't know that, you might write that down. Myelinated axons propagate APs more rapidly, as we just said. There's two other factors. The diameter of the axon. How big around the axon is. So in other words, how thick, how wide this is. The bigger it is, the larger the diameter it has, the faster it sends action potentials. An example, large diameter myelinated axons, we call these A fibers, can conduct an action potential at velocities ranging 12 to 130 meters per second, which sounds pretty fast, but we don't really know meters per very well in this country. We all know what miles per hour are, right? We can transmit signals at 280 miles per hour. That's incredible. And these carry urgent information such as sensory signals associated with touch, pressure, position of joints, motor signals that cause contraction of skeletal muscles. That's why there's not really a delay between the time you think about contracting your muscle and when your muscle actually contracts. Or when you touch something hot and when you feel that pain, right? It's like instant, right? Okay. Think about how amazing it is that we've got those ions flowing like they do and how quickly that signal travels. Now, on the opposite end of the spectrum, we have small diameter unmyelinated axons. We call these C fibers. They're going to carry signals at 0.5 to 2 millimeters per second, which is only 1 to 4 miles per hour, but that's still pretty dang amazing. Not nearly 280 miles an hour, though. They carry less critical information, things like motor signals that cause contraction of our smooth muscles in our stomach, intestines, and that kind of stuff. And then body temperature, which really doesn't change too much, which is good. And we all know that we're very warm-blooded, which is nice because action potentials propagate faster at our higher temperatures. So that's not an issue really for us, but it is good because our body is warm. So it does promote propagations. Propagation, not shins. Okay, a synapse is the place where signals are transmitted from one neuron, what we call a presynaptic neuron, to another neuron or another cell, what's called a postsynaptic neuron. Do you guys know what the prefixes pre and post mean? Pre means before, post means after. The point of, and I'll use air quotes, contact between neurons is a synapse. And I use air quotes because if you look hard enough, these axon terminals of the presynaptic neuron are not actually touching the postsynaptic neuron. There's a tiny little space in there. Now these are examples of what we call chemical synapses, which seems kind of weird. We'd think electrical because neurons and electrical go together, but these are called chemical synapses. I'm not going to really focus on the types of synapse, axodendritic, axoaxonic, axosomatic, not super important. There's another form of synapse. We won't talk about these until later in the semester when we talk about the heart. Electrical synapses involve action potentials actually just spreading from cell to cell through what we call gap junctions. They're like tunnels between two cells. We have these in our heart, specifically the muscle cells of our heart, which allow muscle cells in our heart to kind of get excited at the same time and then contract at the same time. Something that's super, super important because our heart needs to beat in a very coordinated manner. What we'll focus on for this lecture are chemical synapses. The connection between two neurons, between a neuron and a muscle cell, between a neuron and a gland cell, whatever it is, there's always chemicals that are released, which is why we call them chemical synapses. Okay. There's always a tiny gap. As I said, I use the air quotes, point of connection, or point of contact, I think I said is what I said. That's what I said, that's what I said. I don't know what I'm saying. This tiny gap is called a synaptic cleft, and this requires the presynaptic neuron to release a chemical called a neurotransmitter, which diffuses across the fluid in the synaptic cleft and bind to receptors on the postsynaptic neuron or cell. And these neurotransmitters can be either excitatory or inhibitory. Okay. And in response to those neurotransmitters being released, our postsynaptic neuron or postsynaptic cell, if it's not a neuron, it's just a postsynaptic cell, creates what's called a postsynaptic potential, which is a change in its resting member potential. So it's a graded potential. And so this process involves a presynaptic neuron converting an electrical signal called a nerve impulse or action potential into a chemical signal, which is the released neurotransmitter. Here's what takes place in a chemical synapse. And here's an illustration to show the steps involved. And there are 7 steps, very similar 7 steps to what we saw earlier today. And just like the 7 steps earlier today, you will need to know these steps front to back, side to side, and round, all that wonderful stuff. Same advice I gave you earlier, study the heck out of it. When you feel like you know it, test yourself by seeing if you can write them out on a blank piece of paper. Not something you want to do on the test, but is a good way to find out if you are in good shape. All right, what we are looking at here is the very tip of our neuron. One of those very tips, because remember, there are many branches at the end of an axon, what we call axon terminals. Each of them has a bulby, fattened synaptic end bulb, synaptic knob, whatever you want to call it. Inside of that knob are bubble vesicle, synaptic vesicles is what they're called. Okay. Bubble vesicles, synaptic vesicles, whatever you want to call them, which are full of neurotransmitters. And that's what these little red dots are. Those are the chemicals. So when an action potential comes down the axon, it eventually reaches to each one of these knobs. We're zooming in at one knob, and its point of, and I use air quotes again, contact, because it's not truly contact. There's a tiny little gap called a cleft between it and the second neuron. There's what we call the postsynaptic neuron. Now, it doesn't always have to be another neuron. Could be a muscle cell, could be a gland cell, whatever. It doesn't matter. In this case, it's a neuron. The arrival of that action potential to the knob triggers the opening, step 2, of voltage-gated calcium channels, allowing calcium to come in, which triggers the movement of these bubbles towards the cleft. And then those bubbles fuse with the plasma membrane, and the neurotransmitters are released. Neurotransmitters are the chemicals that bind and can act like a key to open up channels that are on the opposite side of the cleft, in this case, the dendrites of our postsynaptic neuron. So our postsynaptic neuron opens up channels, allowing sodium to come in. And as we know, I go back to this figure. If enough sodium comes in, we can hit threshold, which means, as it says, step 7, we can trigger an action potential in the axon of a postsynaptic neuron. This is how we can take an action potential from one neuron, release chemicals that can trigger an action potential in the second neuron. If I go back all the way to this figure and this example of holding that pen in your hand, got an action potential spreading up that sensory neuron. Neurotransmitters release that trigger an action potential on the second neuron, and then a third neuron, action potential. Signal comes down, action potential. Neurotransmitter release, action potential down. Neurotransmitters release that activate muscle contraction. Think about how amazing all that is for something so simple as just writing with your pen or pencil.
How does the ions are diffusing only a short distance, allowing the signal to move quickly? At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
Overwhelmed yet? Hopefully amazed at the same time though, right? Incredible, incredible, incredible. Everything that goes into one action potential, one release of neurotransmitters, and that's one. I mean, we're talking about incredible amounts every time something happens in our body. Now going back to this slide, it states that neurotransmitters can either be excitatory or inhibitory. which relates to what happens to the second neuron or second cell, either becoming excited or inhibited. Okay. In our last lecture of this unit, we're going to talk about reflexes. Like when you go to the doctor and you sit on the table and they tap on your patellar tendon. And what happens when you tap on the patellar tendon? You kick your leg out, right? Okay. By doing that, and we'll talk about this in much greater detail, we trigger an excitatory signal, which causes muscles of the quads to contract. Because your quads are the muscles that cause you to kick your leg. At the same time, we inhibit the muscles in our hamstrings, our hamstring muscles. Because those are the muscles that bring our leg back, flex our knee. We can't have our hamstrings and quads contracting at the same time because they cancel each other out. So what we do is we create an excitatory signal to go to the quads and inhibitory to the hamstrings. Okay. Neurotransmitters are either going to make the second cell or neuron more negative or less negative. Okay. If we become less negative, which means we're closer to threshold, which means we're closer to creating another action potential, we call it an EPSP, short for excitatory postsynaptic potential. And that's because excitatory neurotransmitters trigger both sodium and potassium channels to open. But what's most important is the sodium channels open faster than the slow potassium channels. So sodium rushes in and we become less negative. Okay. When you inhibit something, you're trying to stop it from happening, right? An inhibitory postsynaptic potential, or IPSP, gets us farther from threshold. In other words, we're inhibiting or stopping an action potential from occurring. In this case, what happens is inhibitory neurotransmitters cause potassium channels only and or chloride channels to open up. So either potassium leaves the cell or chloride enters it, both of which that are going to cause the same thing, us to become more negative than resting membrane potential. In other words, we're becoming farther from threshold. And thus likely that second neuron is is less likely to fire. Or that second cell or whatever it is less likely to hit his excitatory threshold stage. So a little bit more about neurotransmitters, which are obviously super important. A neurotransmitter can essentially be called any signaling molecule released by a neuron. Some bind to their receptors and act quickly to open or close ion channels in the membrane. Others act more slowly via what we call second messenger systems, influencing chemical reactions inside cells. Either way, the process can be excitation or inhibition of those postsynaptic neurons. To be classified as a neurotransmitter, they must satisfy these three criteria. They must elicit the appropriate response when introduced to the cleft. Okay, when that means they need to travel across the cleft and bind to the specific receptor they're built for. They must occur naturally in the presynaptic neuron. It must be released at the right time when the presynaptic neuron is stimulated. So they must be released when that action potential reaches the knob. Now, a lot of like drugs, whether they're prescription drugs or they're illicit drugs, I'll just say illicit, influence the release of neurotransmitters, and that's how they can change moods and give you those, different types of feelings and that kind of stuff, which I'm not going to talk about, but if you were to take like a drugs and behavior class or drugs and psychology type class, you learn a lot of that. It's really cool. We won't get into any of that stuff. We could go into much greater detail about neurotransmitters. For now, I just want to briefly talk about a couple, one of which I've already mentioned, Acetylcholine, abbreviated ACH, is our most common neurotransmitter for synapses between neurons outside the CNS, as well as what we call the neuromuscular junction, which is the connection between a nerve and a muscle. It is an excitatory neurotransmitter at some synapses, it's inhibitory at another, or at others. Any synapse using acetylcholine as its neurotransmitter is called a cholinergic synapse, which the word choline is literally in acetylcholine, so it should be pretty easy to remember that. There's also 3 what we call catecholamine neurotransmitters: dopamine, norepi, which is a lot of times just abbreviated NE, which you might put just so you know it, and then epinephrine, which is a lot of times just abbreviated either E or epi. Of those 3, we really kind of focus in this class on norepi the most. Plays roles in arousal, dreaming, and regulating mood. When we talk about epinephrine in this class, mainly we're going to be talking about it as a hormone being released into the blood instead of being released by a neuron. Anybody know what epinephrine is best known as?
The EpiPen.
It's what's in an EpiPen, absolutely.
It's like if you're having like an allergic reaction, it stops it.
Yep, that's why you take an EpiPen. Epinephrine is just a fancy word for adrenaline, which we all know we get when we have that fight or flight response, right? Talked about that the other day. Both norepi and epi are going to serve our body as hormones, which means that they're released by your adrenaline, or from your adrenal gland, I should say. And instead of being released by a neuron, they're released by your adrenal gland into the bloodstream. Brain neurons contain the neurotransmitter dopamine and are active during emotional responses, addictive behaviors, and pleasurable experiences. You hear dopamine kind of described as like a feel-good chemical. It's our kind of like feel-good natural drug, if you will. Dopamine-releasing neurons also help regulate skeletal muscle tone, which is a form of contraction that we can't even control in our skeletal muscles. It's really, really weak, even taking place when we feel like our muscles are relaxed. Dopamine also helps regulate other aspects of muscle contraction movements. Unfortunately, people with Parkinson's disease, which is an autoimmune disease, causes muscle stiffness, and which is a really kind of terrible way. I need to really kind of change that. Terrible way to describe it. It is because of the neurons in the brain that release dopamine degenerate. In other words, the muscles end up just contracting on their own, and you really don't have much control over them. Michael J. Fox, do you guys know who he is? Probably the best known person with Parkinson's. Really affected him super early compared to what most people get it at. Kind of put his whole career, he was a very famous actor in the 90s and 80s. Might have kind of derailed his career. He's still around, but not nearly as prevalent as it used to be. Synapses using catecholamines as their neurotransmitters are called adrenergic synapses, and we find them at junctions between nerves and between muscles and our internal organs, as well as nerve-to-nerve junctions in the brain. Last thing here on neurotransmitters. We said earlier in this lecture that we store neurotransmitters in those synaptic vesicles, these little bubbles in our synaptic bulb or in knobs. What this picture doesn't show, actually, I'm going to go back to it. When we are in a resting state, these vesicles are held in place by little proteins. They're called docking proteins. And when calcium flows in, it kind of breaks them apart, and that's why those vesicles can move to the cleft. Well, there's 2 familiar and potentially deadly illnesses that can interfere with vesicle docking and then thus the fusion with the plasma membrane. Tetanus toxin prevents the release of neurotransmitters from inhibitory neurons in our spinal cord, resulting in uncontrolled muscle contraction. Now, when you see the word tetanus, what's that trigger thoughts of?
Like when you get tetanus,
Tetanus shot, right? And why do people get a tetanus shot?
so they don't get it.
So they don't get this toxin in their body, which can cause a condition. You've ever heard of lockjaw before? Okay. What this happens when you step on like a rusty nail is that toxin can get in your bloodstream, and it prevents your muscles from being able to relax. And that's why it's called lockjaw. Your muscles in your jaw, or move your jaw, I should say, clench down and you can't open your mouth. Botulinum toxin prevents the release of neurotransmitters from motor neurons, resulting in kind of the opposite. Instead of muscle contraction, it causes weakness and paralysis. You ever heard of botulism before?
Yeah.
Botulism you get from like,
Bad cans, food cans.
yeah, don't get the dented cans, even if they're half off. If you combine Botox there as one word, probably heard of Botox before, especially Lauren, since you have your background. People inject that into their face a lot of times to get rid of what?
Wrinkles.
Wrinkles. What they're doing is purposely injecting and causing facial muscle weakness, paralysis, and so their face looks a little bit better, which is amazing that somebody figured that out. But it's also amazing that people do that because they're paralyzing their face. Now, it's not permanent, but still. It can be deadly if you get it in your respiratory muscles and then you can't breathe because you can't contract the muscles that allow you to suck the air in. Okay, any questions? I know we were supposed to start the next lecture, but that's okay. This was a long, very technical, very important lecture, so I wasn't trying to race through it.
What time are you gonna be here tomorrow?